Hello everyone, today we're going to look at chapter 4 from the IGCSE Biology syllabus, which is biological molecules. So we will look at different types of biological molecules, but first we need to understand the concept of monomer and polymer. So for different type of biological molecules like carbohydrates or proteins, the basic building blocks of those biological molecules are called monomers. And when those building blocks are bonded together to create a larger molecule, we call that larger bonded molecule polymer. So monomers build up to make a polymer. This is the underlying concept of all biological molecules. So first biological molecule we will look at is carbohydrates. So carbohydrates are molecules that are only made up of carbon, hydrogen, and oxygen. So it could be a simple sugar, or two sugars bonded together, or lots of sugars bonded together. So the simple sugars are called monosaccharides, and complex sugars are called polysaccharides. So they are all carbohydrates. So for simple sugar, we have glucose, for example, and when those glucose are bonded together, it could form a glycogen or starch or cellulose in plants, which are all complex sugars. The next biological molecule we will look at is fats or lipids. So fats are made from glycerol and fatty acids. And similar to carbohydrates, it is mainly made out of carbon, hydrogen, and oxygen. So the glycerol and 3 fatty acids are the monomers or the building blocks of fats. And the lipid molecule, where glycerol and 3 fatty acids have combined, is the polymer, yes. We usually call that single lipid molecule triglyceride. And we will also look at protein. So proteins are made up of amino acids. And amino acids have carboxyl parts, which contains carbon, oxygen, and hydrogen, and also the amine parts, which includes nitrogen and hydrogen. So proteins are mainly made out of carbon, hydrogen, oxygen, and nitrogen. So different sequences of amino acids, the molecules shown here, give different shapes to protein molecules. So when these series of amino acids are joined together, so the amino acids are the monomers, they form a protein, which is a polymer. So for example, when they form different proteins, it could be an active site of enzyme or the binding site of the antibodies. So we looked at 3 basic biological molecules, and we will move on to food test, more specifically how we will detect if certain biological molecules are there in the solution. So for detecting the presence of starch, we use an iodine test. So just simply you have to add an iodine solution to a solution which you want to know. So the color of iodine solution would change from red-brown to blue-black when starch is present. So if starch is not present, the solution would continue to stay in red-brown, which is the color of the iodine reagent. The next test we will look at is Benedict test. Benedict's test is used to test the presence of simple sugars, so the single sugar molecule like glucose. So the procedure is a little more complex compared to the iodine test. The first, you have to add the Benedict solution or the Benedict reagent, and you need to boil the solution in a thermostatic water bath, for example, over 60 degrees Celsius. And then after cool down, you have to observe the color change. So if the color of Benedict solution changes from blue to brick red, it indicates that simple sugars or glucose are present. And if the color does not change and stays in blue, it means that simple sugars are not present. So it may be that glucose are not there at all, or only complex carbohydrates or starch is there. The third test we will look at is Burette test. So Burette test is used to test the presence of proteins. So it has a simple procedure, just adding a Burette reagent. And the color of the solution would change from blue to purple, or more better wording is lilac, when proteins are present. So if protein is not present, the solution would continue to stay in the blue color. And we will also look at ethanol emulsion test. So ethanol emulsion test is used to test the presence of lipids or fats. The procedure is that you dissolve the solution you want to test inside the ethanol and shake it. And then you move the mixed solution into a cold distilled water. And after some time, you will observe for a milky white emulsion. So when lipids are present, a cloudy emulsion will appear on the top of the test tube, for example, if lipids or fats are present. So, and conversely, if lipids are not present, the solution would continue to stay colorless, even though you shake it and add more ethanol. Yeah. And finally, you have to also know how to test for vitamin C. Sorry for the mistake. It's supposed to be vitamin C. So we use DCPIP test for vitamin C. So the procedure is just add DCPIP solution. And DCPIP solution has a blue color. And the color of the solution would change from blue to colorless when vitamin C present. You will just see how the color of the solution fades. However, if vitamin C is not present, the DCPIP solution will continue to stay in the blue color. So to summarize, we first have iodine test for starch, which yields red-brown color for negative results and blue-black color for positive results. Positive results means that the molecule is present. So then Benedict test we use for testing simple sugars. The negative results is blue and the positive results is brick red. And Buret test for proteins, blue for negative results, lilac for positive results. Ethanol emulsion test for lipids, colorless for negative results, and cloudy emulsion appearing for the positive results. And finally, this is a PIP test for vitamin C, and the blue color will stay for negative results, and the blue color will fade to colorless for the positive results. And then there is another important biological molecule we have to understand, we have to learn. It's the DNA, or the nucleic acid. So DNA is made up of chemicals called bases, or nucleotides. So as you see here, these colored blocks inside the DNA are called bases. And two strands of bases coil together to form a double helix structure shown here. So this one long backbone of phosphate, so the long backbone and bases attached to that backbone is called a single strand. And there is two of these strands bonded together to form a double helix. So the strands are linked together by hydrogen bonds, which you will learn later, between pairs of bases. You just have to remember that there are bonds between pairs of bases that link the strands together. And one interesting rule you have to understand is that so there are 4 types of bases: adenine, thymine, cytosine, and guanine. You only have to know the initials A, T, C, G. And you have to remember that A only binds with T and C only binds with G. So that's all for DNA. Let me add one more thing. So DNA has this phosphate backbone, and inside adenine, thymine, cytosine, guanine, they are nitrogenous bases, so they contain nitrogen. So DNA is mainly made out of carbon, hydrogen, oxygen, nitrogen, and phosphorus. So to summarize it, carbohydrate is only made out of carbon, hydrogen, oxygen. Same with lipids. And proteins are made out of carbon, hydrogen, oxygen, and nitrogen. And finally, nucleic acid is made out of carbon, hydrogen, oxygen, nitrogen, and phosphorus. And one final thing you have to remember is that water is really important as a solvent to digest, transport, and excrete these biomolecules, because these biological molecules are dissolved inside the water to be transported or excreted or absorbed into our cells. And that's all for 4.1, and thanks for listening to the lecture today.